Welcome to RFD-TV Live, an informative and interactive hour featuring topics and products of interest to rural America, with expert guests on hand to answer your questions or discuss your feedback. And now, RFD-TV Live.
Good evening, everyone. My name is Mike Adams, and I'll be your host for tonight's RFD-TV Live panel discussion. Now, some of you may know me as the host of the nationally syndicated radio talk show AgriTalk, where we discuss issues and ideas important to rural America. In the next hour, we'll be doing that as well. We'll be discussing a major breakthrough in soybean research. You know, it wasn't that long ago that research was done to map the genome of a human being to better understand all of our genetic makeup and move forward with development in medicine and better understand genetic diseases in humans. Well, this same fundamental research has now been accomplished for soybeans. Let's watch.
The expectations of the soybean market have changed greatly in the past few years. Today, farmers expect more from their seed. Herbicide resistance, drought resistance, advanced oil traits. Seed breeders are charged with packing all these important traits into the soybean seed quickly and effectively. That's where the efforts of the researchers mapping the soybean genome sequence come in. With the help of the United Soybean Board and Soybean Checkoff-funded genomics tools, the U.S. Department of Energy Joint Genome Institute has mapped and released a draft of the soybean genome sequence. But what exactly is the soybean genome sequence?
One of the analogies you can use about the genome sequence is it really gives you the parts list. Based on the sequencing that was previously funded by the United Soybean Board and other work that had been done, we knew there was about 36,000 genes in the soybean genome, so you can think of those as 36,000 parts. With the full genome sequence, we now know there's over 65,000 genes. So now we have a complete part list. The soybean genome sequence is kind of the beginning. What it does is it enables a lot of other technology to be applied to soybean. An example would be oil-protein relationships in the seed. Farmers know that as you drive up oil, you drive down protein. If you drive up protein, you drive down oil. And yet both oil and protein are very valuable. So what we'd like to do is create a soybean that has both high oil and high protein.
Thanks to Checkoff-funded research and other work, we now have a better understanding of a soybean's genetic makeup. But where do we go from here?
Knowing the genome sequence really helps basic research and also applied research. One of the things that we'll do is allow us to attack a lot of the agronomic problems in a much more precise way. You know, many years ago, soybean breeders only had to worry about things like yield. But now we're getting much more sophisticated. We need to modify the oils in the soybean seed. We want herbicide resistance. We want soybean cyst nematode resistance. And as we bring more of these traits into a particular genotype, we have what's called linkage drag. So we bring in a gene for, say, soybean cyst nematode. We might also bring in a gene for low yield. Well, we don't want that. So having the genome and being able to design, for instance, markers that allow us to do our mapping and our breeding much more precisely will help eliminate some of those problems. I think it's going to enable a lot of applied research and allow us to do it quicker in a much more precise way. For instance, in the past, we knew there were genes for, say, resistance to soybean rust, but we actually didn't know what those genes were. Now with the genome sequence, we can actually go in and actually identify those genes. And recently, researchers at Iowa State University actually succeeded in doing that and actually identifying a gene that determined soybean rust resistance. That allows then much more precise markers to be developed. It also enables biotechnology approaches to actually increasing soybean rust resistance. So, and I could go on. I mean, the soybean genome sequence is tremendously enabling for all kinds of both basic and applied research.
But what does this groundbreaking research mean for the soybean farmer?
I think the soybean genome is really going to allow us to attack a lot of the problems that the soybean farmers are concerned about, such as drought, biotic stress, fungal disease resistance, soybean cyst nematode resistance. I think they're going to see some rapid progress now in at least the understanding of those diseases and new strategies by which we might be able to attack those problems. Right from the very beginning, it was the plan of the soybean community that whatever information was obtained, that information should be immediately made public as quickly as it was confirmed. And that's been the policy right along. If you make the information publicly available, it means anybody can use it. And that means that the progress, the use of that information is going to be disseminated very broadly and put to use very quickly.
So, what made this breakthrough project possible in the first place?
7 or 8 years ago, the United Soybean Board actually funded the initial sequencing project. In that case, we were sequencing individual genes and not the whole genome. But that then gave us the confidence and gave us the beginning data that we needed to convince people that sequencing the genome would be possible. USB was also very much involved all the way along, for instance, in sponsoring community workshops and community meetings. And that was very critical for getting the soybean research community together, which included not only the basic researchers, but also breeders, and also industrial involvement. So getting that cohesive community using USB funding was very important. I'd say I think all the researchers, all the soybean researchers in the country are very grateful to the soybean farmers. We have a very progressive group of farmers. I mean, when we go and talk to them about the importance of the soybean genome sequencing, it's not a hard sell. They see the relevance right away, and they've been extremely supportive. And I think, again, that's a reflection of how the soybean community is cohesive and is behind these kinds of efforts. What USB has done through the checkoff and also the state checkoff boards is that they provided the initial kind of pilot project money that allowed us to show proof of principle, and allowed us to convince people that in fact it was possible.
So now you have an idea what we're going to be talking about tonight. This is very exciting technology, a very big breakthrough for the soybean industry. We're going to try to explain how it works and why it is important. Let me introduce our panel for you tonight. With us first is Rick Stern. He's the current chair of the United Soybean Board's Production Committee and a soybean farmer from Cream Ridge, New Jersey. He'll be discussing how the soybean checkoff helped make this immensely important research happen and what it means for America's soybean farmers. Rick, thank you for being with us. And I didn't realize New Jersey was such a hotbed for soybean production, but you grow a few soybeans out there, don't you?
Yeah, we definitely have a few soybeans grown in there, and thanks for having me here tonight.
Very good. Look forward to talking with you. Also with us is Dr. Roger Borma from the University of Georgia's Center for Applied Genetic Technologies. Dr. Borma is a soybean breeder that applies these new technologies for the improvement of soybean varieties. Thanks for being with us.
You're welcome, Mike. Glad to be here.
A lot to talk about with you and our other researcher that is with us here, Dr. Perry Cregan of USDA Ag Research Service at Beltsville, Maryland, who is one of the genome researchers involved in this process that we're talking about tonight. He'll be telling us what the implications are for soybean research and soybean improvement thanks to this breakthrough. Dr. Cregan, thank you for joining us. Let's start right there. Now, mapping has been going on for a number of years now. How is this different than what we've been doing up to this point?
Well, what we've done over the years, and this is beginning with checkoff support actually back in the mid-90s, is develop soybean mapping, which is identifying points along those chromosomes of this DNA sequence that are associated with genes that control various traits for productivity, for disease resistance, for quality, and then using those markers as surrogates for selecting genes that will improve the soybean in some way.
Now, when you say